Well, hello everyone, and welcome to the Centre for Eye Research Australia's 13th annual Gerard Crock Lecture. My name is Keith Martin, and I'm Managing Director of CIRA. So before we begin, I'd like to acknowledge the Wurundjeri and Bunurong people of the Kulin Nation, the traditional owners of the land where I am now, and the Kaurna people as custodians of the Adelaide region, where our guest lecturer, Professor Robert Casson, is tonight. And I pay my respects to their elders, past, present and emerging, and to their families. So tonight is the 13th annual Gerard Crock Lecture, and for me personally, it's the 4th I've had the pleasure to be involved with. And the Gerard Crock Lecture series honours the memory of a renowned ophthalmologist, the late Professor Gerard Crock, inaugural Ringland Anderson Professor of Ophthalmology at the University of Melbourne. We've all had a turbulent couple of years, and this lecture is one of our events that's been majorly impacted by the pandemic. So I'm really thrilled and very thankful that many of you could still join us tonight for such a special event in the CIRA calendar that honours Professor Crock's memory. So whilst it's disappointing that we can't welcome you in person tonight, it's great to see so many of you still embracing online events. We've had rather too many of those over the last couple of years, but we're all getting pretty competent at it now, I hope. And we do hope that we'll be able to welcome you all in person in the very near future. So Gerard Crock was always looking for ways to make a difference to patients. And as Australia's first professor of ophthalmology, he was an innovator in eye research and an early adopter of many different types of technology. And by all reports, he was an incredibly skilled surgeon and was involved with saving the sight of thousands of patients over the years. And as I mentioned earlier, Gerard Crock was appointed Melbourne University's Foundation Ringland Anderson Professor of Ophthalmology in 1963, and he was only 34 at the time, which is really amazing. And in true pioneering fashion, his was the first medical specialty chair in Australia. And he played a really crucial and overall an enormous part in establishing both University of Melbourne's Department of Ophthalmology and the Royal Victorian Eye and Ear Hospital. So tonight I'd like to welcome all of Gerard's family that are joining us online. We have at least 10, and in particular we extend our thoughts and get well wishes to Gerard's wife Jackie, who's been unwell recently, but I understand is mounting somewhat of a remarkable recovery at the moment, and that's just fantastic news. We've all been thinking of you and sending our best wishes to you and the family tonight. And we at CIRA are also deeply appreciative of the Crock family's ongoing commitment to CIRA and to our research. And indeed, Gerard's family have asked me to express their appreciation to the University of Melbourne Department of Ophthalmology and to CIRA for continuing to advance eye research. His family has no doubt at all that Gerard would have been thrilled to see the progress that's been made in eye research and eye treatments, and especially thrilled to see how the department has grown since its early days as one desk and chair in an otherwise empty room. And I think we have that picture. We may be able to show you that picture now, which was found just very recently again. So in other welcomes, I'd like to send a virtual welcome to everyone joining us online this evening. Welcome to our colleagues from the University of Melbourne and from the Royal Victorian Eye and Ear Hospital, with special mention to the Harold Mitchell Professor of Indigenous Eye Health and former CIRA Managing Director, Laureate Professor Hugh Taylor, and to the Eye and Ear Hospital Chief Executive Brendan Gardner. So CIRA and the University of Melbourne have an extremely strong partnership with the Eye and Ear Hospital, and I've personally enjoyed greatly working closely with Brendan and his team as we work to develop even closer relationships, including a new joint strategy, which I think is very exciting for both of us, joint strategy for the Eye and Ear Hospital and for CIRA. Welcome also to our CIRA board members and research colleagues and many CIRA staff that are joining us online tonight. And of course, welcome to all of our generous supporters and donors who make much of our research possible. So just as an introduction to CIRA, for those of you who may not be aware, particularly those who are joining a CIRA event for the first time, CIRA is an independent medical research institute, and we're part of a world-leading research community based in Melbourne, Australia, and we enjoy close associations with the University of Melbourne and the Eye and Ear Hospital locally. And our team of basic scientists and clinical scientists is absolutely committed to conducting research that helps to detect and diagnose eye disease earlier and to prevent vision loss, and even hopefully to restore sight. So we use hope in sight as our tagline, and that reflects our optimism for the future of eye research and for what we can achieve for patients experiencing vision loss and blindness. And at CIRA, our mission is simply to eliminate the major causes of eye disease that cause vision loss and blindness and to reduce their impact on people's lives. And at the heart of that is innovation. We're trying to do things differently and do things better to make lives better for the patients that we interact with. So please join me in watching this short video, which focuses on one of our fast emerging areas of research, and that's gene and cell therapy. Eyes are amazing. Eye doctors and researchers can examine the intricate structure of an eye and how it works in more detail than just about any other part of the body. That's one reason why we're seeing so much progress in gene and cell therapy research for eye disease. This research is helping us find new treatments to prevent vision loss and restore sight. Gene therapy is a technique we can use to treat blindness or vision loss caused by defective or missing genes. Our genes make us who we are and influence how different parts of our body work, including our eyes. With gene therapy, we first identify a defective gene causing blindness or vision loss, then we create a correct, functioning copy in the lab. This correct copy is then merged with a modified, safe virus known as a viral vector. This virus can't cause infection, but is very effective at getting genes into cells. Finally, we inject the virus directly into the eye, and the correct gene is delivered to the target cells. Gene therapy can be used to treat a wide range of diseases by delivering genes that help make cells in the eye more resistant to injury. It can also be used to directly edit genes, where we can replace a faulty section of DNA. Cell therapy is an alternative approach that aims to replace cells that have been lost with new, healthy cells. These new cells can either be from a healthy donor, or we can take some of a patient's own blood or skin cells and turn them into eye cells in the lab. This research gives us hope that new treatments are on the horizon to prevent blindness and restore sight. So I hope that helped you to understand a little bit about some of the new things that we're doing at CIRA. So just before I introduce Professor Robert Casson, just some general housekeeping. Tonight we encourage you to ask questions throughout the presentation, and you can do this by typing your question in the Ask a Question box you should see at the bottom of your screen. And if you're watching on Facebook, you can leave a comment underneath the live stream. And tonight's lecture is also being recorded, and everyone who's registered will receive a link so you can watch again or catch up on any bits that you missed. So just to introduce Bob, at CIRA we know that we can't achieve our vision of a world free of vision loss and blindness by working alone. And collaboration is a really important and a really fun part of what we do. We work with many researchers from many different institutes in Australia and around the world, and that's critical to the success of our research. And tonight, we're really delighted to be able to bring you a presentation by one of Australia's most innovative eye researchers, and I think I can say that with an extreme degree of confidence, and a collaborator in many research projects, and a good friend of mine for many years, and a close collaborator, that's Professor Bob Casson. So Professor Casson is the head of the discipline of ophthalmology and visual science at Adelaide University, and he's an internationally recognized clinician scientist ophthalmologist with a special interest in glaucoma, neuroprotection, retinal laser interactions, epidemiology, and translational ophthalmic research. He became a fellow of the Royal Australian New Zealand College of Ophthalmologists back in 2000 and completed his lab-based PhD at Oxford University in 2004. And I think it was during your time in Oxford, Bob, that we first met when you were working with Neville Osborne in the lab there, and we've been in pretty close contact ever since. And Bob completed a biostatistics degree. He's formidable with numbers as well as everything else at University of Adelaide in 2012. So, and along with his work at Adelaide, he's a consultant ophthalmic surgeon at the Royal Adelaide Hospital and a founder and vice chairman of the Adelaide-based charity Sight for All, which trains eye doctors in the developing world. So tonight, Bob will be presenting on using low energy light to treat retinal disease. And we think of light as, you know, what we use to see and what we use to examine the eye. But light is also, as Bob will show us, potentially really interesting therapeutically in terms of a treatment modality for certain sorts of eye disease. So Bob, we're really looking forward to your lecture and hearing what you have to say. But so you're very welcome indeed. But just before we begin tonight's formal lecture, I'd like to have a chat with you about a few different things, Bob, if that's all right. So we'll just let you say hello, and then we'll talk about a few of these things.
Well, hi Keith, and hi to everybody at CIRA and everybody out in cyberspace. Thanks so much for attending this talk. You know, it's a great pleasure to be here. It's a great honour to be invited to be here. So thanks for that.
Thanks, Bob. And I say we're really sorry. We owe you a nice dinner out in Melbourne, which would have been happening after this tonight. And so next time you're up, we'll have to honour that. Bob, as ophthalmologists, one of the frustrating parts of our job is having to tell patients there's nothing more we can do to save their sight. And you know, many things we can fix, but there's still a whole bunch of stuff that we can't fix. And I guess that's why, you know, as researchers, we work hard to change that. Can you tell me a bit about the importance of research for you in your, you know, daily life and daily practice?
Well, I guess from a couple of perspectives then, Keith. I mean, I guess from a personal perspective, I've loved science for as long as I can remember, since I was a little kid. And so I combined that with my passion for ophthalmology, bringing science to ophthalmology to try and improve outcomes for patients and to reduce the burden of visual impairment in general at a population level as well as at an individual level. But I guess on a more general perspective, research is really the engine room for all of our medical progress, isn't it? So, I mean, when we think about the wonderful modern medical technology and the ophthalmic technology that we all enjoy in the modern era, we can imagine or think back to what things were like 100 years ago. Well, they are the way they are now because of research. It forms the foundation for all the progress that we make. So it's absolutely essential to stop us standing still and to improve the welfare of everyone.
Absolutely. And I guess, Bob, the arrival of a nasty little bug has made the last couple of years a bit unpredictable for all of us, to say the least. And you know, credit to the researchers at CIRA and your institute. They've managed to keep things going, and research has continued, and we've seen significant advances in both places. But you know, can you tell us a bit about what the last 2 years have been like for you and the team in Adelaide?
Well, I guess, you know, in South Australia, we were extremely lucky, you know, and we know that some places were more lucky than others. Some individuals were more lucky than others, depending on, really a lot boiled down to what sort of work you did in this period, didn't it? But we were very fortunate here. I guess from a research perspective, the clinical trials were slowed down, but we were fortunate that the laboratory research was mostly able to continue. There was a big spanner in the works in the overseas work we do with our training and activities with the NGO site for all that you mentioned. But we all learned to use new technology like Zoom and other information technology that allows us to kind of keep going and achieve results through different means, even if you can't visit places.
Yeah, I think your carbon footprint has probably improved to a similar degree to mine, Bob, which is probably no bad thing in the long run in some ways. So, you know, there have been some positives, and it has been an exciting time for, you know, the development of new technologies. And you know, what do you think have been some of the biggest breakthroughs recently? I mean, obviously we're going to hear about your work, but what else has caught your interest recently?
Well, if we go back in time a little bit, just as an example, that seems like it's all totally familiar now, is the OCT machine, optical coherence tomography. So many of the people in the audience might have had one of these scans done of their optic nerves or their maculars. This was the very first reports of this technology was in 1991, and they included a an image of a pretty average image of a retina in that report that was published in Science. So that was barely 30 years ago. So that's just an example of the speed that some technology can be adopted. In more recent times, and things, as you mentioned in the video, stem cell technology and gene therapy is really coming to the fore now. So not all that long ago, that was all science fiction, and now it's in the clinics, and ophthalmology is really leading the way in that area. So the eyes, of course, lend themselves beautifully to gene therapy, and outpouching of the brain lend themselves to model diseases of the brain, let alone all the ocular conditions, of course.
Absolutely, and I guess one of the fun things about what we do is being involved in different collaborations, and you and I have been involved in a few different clinical studies. In fact, long before I came to Australia, we were co-investigators on the glaucoma initial treatment study, the GITS study, and I think we were known as the old GITS as the principal investigators as part of that study. And I guess we've got this new study coming online, the vitamin B3 study, which is really exciting. And Flora Hui from our institute's heading down your way in the next few days, and we meet regularly, and with Jonathan Crowston as well, the former director of CIRA, who's also involved in this big clinical study that we're planning, along with collaborators in the Karolinska. So we're really excited about kicking that off as well, and that's an example of how important collaboration is, because we need to recruit more patients that we can possibly get in one place. But I'm interested in how you go about communicating with patients about trials like this, and about the importance of research, and how you encourage your patients to take part in research studies.
Well, I think it starts with having an important question to answer. So something that really needs an answer. There needs to be a gap in the ophthalmic practice. So B3 is a great example, actually, as you mentioned. So the vitamin B3 is a really exciting study. I guess it depends to some extent what you're asking the patient to do as part of the clinical trial, but the B3 is a particularly easy one because all they're doing is taking a vitamin tablet. So, but we find patients are generally very willing and forthcoming to be enrolled in clinical trials. Recruitment can be difficult at times, but I think basically explaining the nature of the trial to the patient, why the study is important, and many patients will give their time and really are willing to be part of something that they feel can make a difference.
I think that's very true, and it's amazing how generous so many of our patients are with their time to contribute to moving things forward, even though when sometimes there's no particular benefit for them personally, but they recognise the need. And every single treatment that we have that's ever been developed has been through a clinical trial, and so people have had to step up, for any of the treatments that we use today, and that applies to everything that's coming for tomorrow as well. So, and I think finally, Bob, I think it's important to recognise that the work we do wouldn't be possible without the support and generosity of our donors and supporters, and I guess that must apply to your work as well.
Absolutely, Keith. So really the government, for instance, funds very little ophthalmic research. Ophthalmic research is a relatively small field compared to some other gigantic fields of research, and we rely a lot on philanthropic support. I know CIRA does too. And without the generous support of donors, really the research would come grinding to a halt.
I think it's absolutely true. Absolutely true. Bob, great to talk to you. We'll have plenty of time for some questions at the end. Again, those of you listening, please do send your questions in. They will come down the internet tubes to me, and if I don't knock over all the devices on the desk here, I'll be able to ask a few to Bob towards the end of the talk. Or at the end of the talk. So over to you, Bob, and we look forward to hearing your 13th, or not your 13th, but our 13th Gerard Crock Lecture, and you're first.
Okay, so thanks so much, Keith, for the very generous introduction, and thanks indeed for inviting me to present the Gerard Crock Lecture, the 13th one. It's a huge honour and a great privilege and pleasure to be with you. The only disappointment, of course, is that we can't all be face to face, but I'm hoping we're turning the corner on that front. So, distinguished guests, members of the Crock family, ladies and gentlemen, this evening I'd like to discuss a topic that my research team has been investigating for several years. We feel it's become increasingly fascinating and with exciting potential applications to the treatment of blinding eye diseases. Keith has already said a few words about Gerard Crock, but I'd just like to say a few of my own. I didn't know Professor Crock personally. He was before my time, but it's clear from his legacy that he had an inquiring scientific mind that spanned many interests. He pioneered fluorescein angiography. This is now, of course, a commonplace technique in modern ophthalmic practice. He developed a binocular indirect ophthalmoscope, and I have one still just like it that I still use. He pioneered electron microscopy in retinal research, and amongst other things, he made important contributions to ophthalmology in the Pacific. It's clear that he had a passion for applying his keen mind to solving ophthalmic problems and improving the human condition in general. Perhaps most importantly, he fostered that spirit in others so that his legacy lives on, as evidenced clearly by the outstanding work that is conducted at the Centre for Eye Research Australia, and indeed across Australia by new generations of ophthalmic clinician scientists. And I like to think that Gerard Crock would be proud of what he sees around Australia in modern times. So I'd like to discuss a topic, as Keith mentioned, that is treating the retina with low energy light. This has become quite a fascinating topic, and we began investigating this a few years ago. It turns out that light in certain forms can affect the energy supply of the retina. And as many diseases of the retina and optic nerve are caused by an impairment of energy supply, this novel therapy represents a possible new treatment to many blinding conditions. But firstly then, as by way of background, I'd like to discuss what is light, what is the retina, and how does the retina make energy? So firstly then we might ask, well, what is light? So in the 1600s, Isaac Newton conducted a series of absolutely brilliant experiments. He was using prisms that he probably purchased from Showbridge Fair in Cambridge, and he showed that white light could be split into multiple colours that he called a spectrum. He coined the term. He decided that there were 7 colours in the spectrum because he thought that the usual Roy G. Biv colours that we know in the rainbow, because 7 had sort of mystical connotations for him. But there is in fact a continuous spectrum, as it were, of colours. And he proposed that the light was made of tiny particles, corpuscles, that were slightly different sizes depending on the colour. And this became known as the corpuscular theory of light. It held sway for quite some time due to Newton's influence, but was challenged by an alternative theory proposed by Christian Huygens, who suggested that light was in fact a wave, not made of anything. It was merely vibrations of the medium that it was travelling through, like ripples on a pond. Newton's ideas dominated for a while, but later Fresnel and others found further evidence matching the data. So the hypothesis from Huygens tended to support the data that was at hand, that in fact light was a wave, and the corpuscular theory found its way into the trash can of ideas. Until early in the 20th century, Einstein provided some revenge for Newton, not that he really needed a lot of revenge, but Einstein showed that light was in fact made of tiny bundles of energy, packets of energy called quanta of energy called photons, and that these photons could interact with atoms. They could in fact knock electrons out of atoms, the so-called photoelectric effect. And it was for that actually that Einstein won his Nobel Prize in 1921, not for general theory of relativity. And with the development of quantum theory in the early decades of the 20th century, light became sometimes considered as a wave and sometimes as a particle, and is really best described mathematically. But a key point for our story is that light itself interacts with atoms. Considered as a wave, the visible light that our eyes can see forms a fairly narrow band in the electromagnetic spectrum. So this is not to scale. So we have X-rays at very short wavelengths and high energy down one end, and infrared and microwaves further down, radio waves even further down. Visible light is a narrow band from about 400 to 700 nanometers, where a nanometer is 1/1,000,000th of a millimeter, with blue light at the shortest wavelengths and red light at the longer wavelengths, corresponding to lower energy levels. Let's take a brief look at how light interacts with the eye. So the eye really is a marvel of nature. It is, of course, our aspect to all that we see and all the beauty in the world. And it is also a window to the brain and to many of the afflictions that we suffer from. Light is focused by the cornea and then the lens onto the light-sensitive film at the back of the eye, the retina, where it's converted to electrical signals that are then transmitted along the optic nerve to the rest of the brain. So let's take a look in a little more detail at some of these photons as they strike the retina. So the retina is a beautiful layered structure in all vertebrates. It's very similar. And incoming photons, there they come, strike these cells at the back of the retina called photoreceptors. Now, we have two main types of photoreceptors. The rods are super sensitive to photons and are responsible for our night vision. Cones, the other type of photoreceptor, are tightly packed at the center of our retina at a region called the macula, and are responsible for our fine vision, for seeing faces and for reading and driving and most daily tasks and so on. Light striking these photoreceptors triggers a cascade of reactions, chemical reactions that then transmit signals downstream to intermediate cells known as bipolar cells, then to retinal ganglion cells at the front of the retina. And the fibers of these retinal ganglion cells comprise the optic nerve, which then transmits messages to the brain. Now, all of that requires a lot of energy. So that really is a key message. In fact, all biological processes require energy, some more than others, and the retina requires a lot. Let's take a bird's eye view of how life harnesses energy. This is the famous pale blue dot photograph captured by Voyager 1 when it was about 3.7 billion miles away, out beyond the orbit of Neptune, heading off into interstellar space. And one of the lead scientists on the Voyager project was Carl Sagan, one of my heroes. And Carl gave instructions to turn her around one last time and take a look behind and snap a photo of where she come from. And this was the image, or one of the images at least, that it took. This dot, this speck here, this mote of dust, as Carl called it, is Earth. As far as we know, the only place in the universe that harbours life. And when vision evolved, that mote of dust, as far as we know, is the only place in the universe that can see the universe. Now, the human retina is the product of billions of years of evolution of life on Earth, but old habits die hard. And the secrets to the retinal energy metabolism were forged in the earliest period of life on Earth. Let me tell you a brief story about that. So this is not Mars or some exotic exoplanet. This is a NASA artist's rendition of the early Earth at about 4.5 billion years ago, shortly after formation, before oceans, and when the atmosphere comprised mainly carbon dioxide, water vapor, and volcanic gases, almost no oxygen and no life. So the origins of life remain mysterious, of course, but it's certain that for life to exist, it needs to extract energy somehow from the environment. Now, energy can neither be created nor destroyed, but it can be transformed, and it exists in various forms such as heat, light, electrical and chemical energy. Now, it turns out that all life on Earth uses the energy from chemical reactions. And all of chemistry is really about the interaction of electrons. So here are two atoms with their central nuclei, consisting of protons and neutrons. And in orbitals around the nuclei are electrons whizzing around, and they like to hang out in regions where it doesn't take too much energy to hang out in. They like to find places where they can hang out where it requires less energy. And so they might come across a nearby atom with a cozy looking place to hang out and decide to jump ship. So this atom would be termed the electron acceptor. This electron moves from the electron donor to the electron acceptor. And that would be a chemical reaction. And chemical reactions release energy to fuel life. As electrons move down their energy gradients, energy is released, and life has learnt to capture that. Life needs to capture energy and move it around to where it is needed. And it does that by storing energy in this molecule here called ATP. Now, it's quite incredible to think that all life on Earth, from bacteria to humans, elephants, tigers, everything in between, requires ATP, has ATP in their cells to move energy around. Now, one idea about the origin of life is that it formed near hydrothermal vents on the ocean floor like these, and that it learnt a really neat trick from the hydrothermal vents to utilize the energy of electrons moving down their gradients to pump protons across a membrane to run through another molecule to make ATP. And the very first creature, the last universal common ancestor of all life on Earth, had ATP, and it's absolutely essential still to keep our retina functioning and to keep us seeing. Now, fast forward about 2 billion years and critters like these evolved. These are cyanobacteria, or blue-green algae. Now, they have a great trick whereby they use sunlight to break down carbon dioxide and water into sugar and oxygen. That's oxygenic photosynthesis, same as modern plants. Now, oxygen released by the early cyanobacteria, these guys were in the oceans, reacted with the iron in the oceans to form rust, which eventually sank to the ocean floors. And over time, a long time, as you might imagine, all the iron in the oceans was used up and oxygen started to fill the atmosphere. And that event is known as the Great Oxygen Event, which began about 2 billion years ago or so and was actually catastrophic for most of the primitive life on Earth, because oxygen is an excellent electron acceptor and is highly reactive, and it was toxic to almost all life on Earth. But for some bacteria that were present at the time, fortunately, it wasn't toxic and life was resourceful. Now, in 1967, the Summer of Love, Lynn Margulis, who happened to be the first wife of Carl Sagan, as it turns out, small world, proposed the idea of endosymbiosis. This is now essentially agreed upon by just about everybody. And the idea is that about 1.5 billion years ago or so, a small creature that could use oxygen to make energy was engulfed by a larger creature, still just a single-celled creature. And instead of becoming lunch, this thing was incorporated into the cell and was used as the cell powerhouse. So this hybrid creature, the biochemist Nick Lane has a great term for it, describing it as a hopeful monster. So this monster became the ancestor of all complex life on Earth, including us. And in fact, the little creature became our mitochondria in virtually all the cells in our bodies, and it has its own DNA, so it still even has its bags packed. And this little creature, an organelle inside our cells now, combines with glucose that we eat and oxygen to make ATP as an energy source. Now, our brains require a lot of oxygen. That's obvious, of course, because a few minutes without oxygen and we're dead. As it turns out, our retinas require even more oxygen per gram of tissue, even more energy hungry than the brain. The retina really is like an absolutely super burning furnace that needs heaps of fuel all the time. Now, this is nicely illustrated, in fact, by the phenomenon of blackout that jet pilots experience under high G-forces, where there is loss of vision preceding loss of consciousness. So this is a simulation of a fighter pilot experiencing G-forces, loss of vision, comes out of the turn, G-forces relax and vision returns, but before loss of consciousness. So the point is that the retina is more sensitive to the impaired flow of oxygen than the brain. Now, some parts of the retina require even more oxygen than others. This slide I've borrowed from some excellent, or adapted from some excellent work done in Perth by Bill Morgan, Steve Kringle and others, where they measured oxygen consumption in the retina. And the dips here are areas in the retina that are consuming a lot of oxygen. So these photoreceptors are consuming a lot of oxygen, and the connections of these retinal ganglion cells are also consuming a lot of oxygen. Now, just a bit of biochemistry here. Don't be too scared with this. Glucose from our blood enters cells, It's converted to another molecule called pyruvate by a process called glycolysis, that in the absence of oxygen, so let's say in sprinters who are running out of oxygen, lactate's building up in their legs as pyruvate is converted to lactate. In plants, lactate is replaced by ethanol, and that's fermentation. That requires this enzyme here, lactate dehydrogenase A, converts pyruvate to lactate. It makes this other substance here, NAD, and this keeps glycolysis ticking, but making a small amount of ATP each time. A kind of reverse enzyme converts lactate back to pyruvate, LDHB. Pyruvate, this blue thing's our mitochondria. Pyruvate enters the mitochondria, splits this, it's got 3 carbon atoms, splits off a carbon, becomes carbon dioxide. That's why we breathe out carbon dioxide. Splits off some electrons that get sucked towards oxygen, the electron acceptor, and releases ATP in the process. Now, we recently calculated that, in fact, the retinal ganglion cells require about 500 million ATP molecules per second to barely maintain any light perception. So the retina, you can see that the retina itself is consuming requires enormous amounts of energy. Now, this is Glyn Chidlow, one of the senior scientists in our lab. Glyn is perhaps the world's foremost expert on a technique called immunohistochemistry, retinal immunohistochemistry in particular. And he's used this technique to try and unravel metabolism at the cellular level within the retina, to try and figure out which cells prefer to eat what. Now, these cells here are retinal ganglion cells, and they have a lot of this LDHB molecule, which, remember, converted lactate back to pyruvate, so that pyruvate could shuttle into mitochondria and be used through an oxidative process. So probably retinal ganglion cells prefer to be receiving oxygen and using their mitochondria. As opposed, for instance, to these interesting cells here are the bipolar cells, those intermediate cells. They have a lot of this LDHA. They convert their pyruvate to lactate and don't really care too much about the mitochondria. These guys, perhaps because they're living in a zone where there isn't so much oxygen in the retina, they are quite happy glycolytically. So this sort of methodology has given us a lot of insights into retinal metabolism, and it's ongoing work. The photoreceptors, as it turns out, seem to make energy any which way they can. They have mitochondrial enzymes in abundance, and they also have glycolytic enzymes in abundance. So they are adept at making energy either through glycolysis in the absence of oxygen or through mitochondria in the presence of oxygen. Now, it's interesting that the experiments I showed you there were applied to vertebrate retinas, but it turns out that invertebrates' retinas are also highly sensitive to oxygen deprivation. Fairly recently discovered, actually, that squid and lobsters, for instance, when oxygen levels are reduced in the water, rapidly become blind. Now, the next key piece of the puzzle is the laser. So in 1957, a young PhD student by the name of Gordon Gould was thinking about the theoretical work of Einstein and light interacting with atoms, and he coined the term in his notebook, light amplification by stimulated emission of radiation, which later became known as laser. 3 years later, Ted Mayman, working pretty much single-handedly at Hughes Laboratories, developed the first working laser. And it was absolutely incredible how quickly this was adopted into medicine and most rapidly of all into ophthalmology. The use of light as a medical therapy has been a concept really since antiquity. Niels Finsen, interestingly, won the Nobel Prize in 1903 for his discovery that blue and ultraviolet light cured a horrible disease called lupus vulgaris, which was cutaneous TB, often affecting the face, a disfiguring condition. And this was a remarkable treatment that he developed. In the 1960s, as our story continues here, Andre Mester was using newly developed lasers at low energies and noted that they had remarkable wound healing properties. Now, light then in the far red range or in the near infrared used for therapeutic purposes became known as photobiomodulation. An unfortunate development in the history of photobiomodulation, in my opinion, is that it has been reported to treat almost every affliction under the sun, from baldness to arthritis and everything in between. And some of the research, let's say, in the literature, in some quarters, has limited scientific rigor. But nevertheless, there is also some very high quality reports in the literature, and it's becoming an increasing field of interest. Now, there's good evidence that photobiomodulation with deep red light acts on the mitochondria in cells. Now, let's just look at this in a little bit more detail now that we know all about chemistry. So here is the cell, is the mitochondrial membrane, and electrons are moving down their energy gradients, being sucked towards oxygen. The final step in this process requires a copper atom. It's catalytic for the final reaction that reduces oxygen to water. And as it turns out, wavelength of 670 nanometers increases the catalytic activity of this copper atom at the subatomic level, really at the quantum level. This enzyme activity is increased and more ATP is produced. Now, in our lab, at the more macroscopic level, we definitely find that light at 670 nanometers activates mitochondria. The pinkish color indicates activation. And we also find an increased activity of the enzyme, this enzyme itself, in cell cultures treated with 670 nanometer light. Now, a few years ago now then, LX Lasers in Adelaide, they've recently sold out to a French company, but Lumibird, but the plant still exists in Adelaide and all the staff are still there. They made a laser, they basically converted a routine photocoagulator laser to produce a 670 nanometer light. And we started collaborating with them to assess the effect of this new laser in the lab. I must confess that I was extremely skeptical of this at first. I had heard reports previously of photobiomodulation from other Australian researchers who had used a light-emitting diode type lasers and already reported some effects. But it seemed a bit far-fetched to me. How could a red light actually be doing anything at all? But early experiments from one of our PhD students did in fact show that it was definitely doing something. It was protecting retinal cells, generalized retinal cells, from bioenergetic impairments. So that spurred us to keep on going with some experiments. And we felt that it would be very important to understand the therapeutic window of this new laser. How much laser did you need to apply before you could actually damage the retina? Now, this work was done by one of our master students, Jack Au. He's now a registrar. And he was treating rats, some non-pigmented and some pigmented rats, because retinal pigment is a key determinant of laser uptake and whether the laser might have biological or in fact pathological effects. Now, at various energy settings, in fact, as high as we could turn the energy setting, in non-pigmented rats, there was no damage to the retina. But in pigmented rats, some pigmented rats at quite high energy settings, there was definite damage to the retina. In a way, this was sort of reassuring to me that it did have a genuine therapeutic index and was actually doing something. It would have been just a stupid finding to find that regardless of how high you turn the energy setting up, it did nothing. So that actually makes it seem like a more genuine type of effect. Around the same time, our colleague and friend, Mark Gillies in Sydney, also had one of these lasers. As far as I'm aware, there's still only 3 in existence. Mark has 1 and we have 2. Mark has a mouse model, genetic mouse model, where the retinal vessels are leaky. And these are fluorescein angiograms of the mice, thanks to Gerard Crock. And this white fluffy stuff here is leakage from the vessels. And Mark was finding that treatment with the photobiomodulation reduced the amount of leakage. And he proceeded quite quickly to a small human study in diabetic maculopathy, where there is leakage from blood vessels at the macula and swelling of the macula. We joined Mark in this clinical trial, demonstrating then that the laser treatment, the patients received 2 to 3 treatments a week for 4 weeks, reduced the retinal thickness, reduced the diabetic macular edema. Now, this was a small study and there's still a lot of work to do yet, but nevertheless, this was motivating. We then turned our attention to this condition, retinitis pigmentosa. This is a group of diseases. It refers to a group of inherited retinal diseases that are a leading cause of blindness in the working age population. Most forms are due to a genetic defect in the rods, the cells that are responsible for our night vision. And so one of the first symptoms that manifests is night blindness. Later, the field of vision constricts, and then eventually, in most people, the cones at the macula also begin to degenerate, but they generally don't express the gene defect. So it's somewhat of a mystery why they degenerate, but there is considerable evidence now that it's due to a kind of bioenergetic failure. So we felt that the photobiomodulation treatment was an ideal therapy to try to see if it could prevent secondary degeneration of cones in individuals with RP. Because obviously, although it wouldn't preserve the rods, preservation of the central vision would obviously be quite a breakthrough. Interestingly, the jet pilot G-force experience provides quite an uncanny resemblance to chronic visual loss in RP. Here's a slowed down version of what the pilot experiences and really what patients with RP experience over the course of decades, with a dimming of the vision, constriction of the peripheral visual field that progresses in towards the center, and then eventually the central vision is snuffed out in most patients. So preservation of that central vision would obviously be quite a thing. Now, John Wood is another senior scientist in our lab. He is a world expert on retinal cell cultures. And John cultured some photoreceptors. They're quite difficult critters to culture, but we demonstrated that the photobiomodulation light protected photoreceptors against bioenergetic impairments and from oxidative stress, another feature of RP. We then moved to a rodent model of RP in mice, genetic defect in mice causing loss of rods, quite an aggressive model with marked loss of rods in early life in the mice, with subsequent degeneration of the cones. Now, these little tiny red dots are the cones, and in animals not receiving any light therapy, they haven't got many cones left. But in those receiving the light therapy, they've got a lot more cones. We repeated this experiment a number of times. It was protecting the various forms of cones that the mice had up to day 60 of their life and even up to day 90 in later experiments. There was an increase in gene expression of the photoreceptors, as we might expect, as there's more photoreceptors. And there was also a reduction in oxidative stress markers in the retina. So all this was quite encouraging. And we felt, given the safety profile of this laser therapy and the need for treatment, we progressed quite quickly to a small clinical trial in patients with RP. So this young lady has RP. She also tragically is deaf, and she only has a small amount of central vision remaining. All the patients in our study had some impairment of central vision. We treated 6 patients at a lower dose, and when safety was proved, we then treated another 6 patients at a higher dose. Patients received 2 treatments a week for 4 weeks. The primary outcome was safety, but we're obviously interested in any sort of efficacy as well. And the secondary outcome then was the change in visual acuity 4 weeks after the final laser treatment. Now, on average, there was a 5-letter improvement in visual acuity. So this was all really quite exciting and definitely motivates further research. Now, we're not the only ones interested in this sort of therapy. Now, this is not a Dalek, although when I drew it, I realized it looked like a Dalek. It's a light-emitting diode source that is also emits 670 nanometer light, and it was used in this study called the Light Sight study, published fairly recently. It was a randomized controlled study. It wasn't a huge sample size, but it was conducted in Europe, and it was used to treat dry AMD, which essentially has really extremely limited treatment at the moment. And they found on average a 4-letter improvement in visual acuity. They went on then to conduct the Light Sight 2 study, which hasn't been published yet, but I've seen some reports of it, also reporting an improvement in visual acuity. And on the back of those findings, as I understand it, this therapy has now been approved by the European Agency for Treatment of Dry AMD in Europe. There is now a much larger LightSight 3 study being conducted in the United States. Now, not all trials in this area have been successful though. So here's an example of one that was not. This is a fairly recent study done by a very respectable group, conducting a study very similar to what Mark and I conducted, testing photobiomodulation in diabetic macular edema. But here, in my opinion, the protocol was really not well considered. They used an LED source of light that was on an eye patch and delivered through a closed eyelid with a very low dose of light. And really, when you do the calculations, hardly any light reaching the retina, homeopathic intensities of the retina. So I think it really demonstrates the need for further research and understanding of what the optimal doses are with this sort of therapy. So I feel that this is an exciting field of research and one that certainly deserves more attention. Future directions for us are to determine the optimal laser parameters for RP, because we really haven't done that yet. We were fortunate to receive an NHMRC grant to do just that, a development grant in conjunction with Alex, to determine what the optimal frequency of delivery is, how long the effects last, and a number of other parameters, to then proceed to a larger RP study, larger clinical trial. We'll be trialling it this year in experimental glaucoma with treatment to the optic nerve in mice and rats with experimental glaucoma. And we have ideas of trialling in other retinal diseases where there's bioenergetic failure, such as retinal vein occlusions. So I feel it's an exciting area of research that is a potential adjunct to current treatments. In terms of funding support, I'd like to thank the RIA, now Australian Vision Research, for support, the NHMRC, and also, of course, to our donors. Thank you very much. And thank you all very much for your attention. Thanks for watching.
Well, that was a spectacular talk. And, you know, I can safely say you're the first Crock lecturer to start your lecture 4.5 billion years ago. I mean, not even Hugh Taylor's managed that. So sorry, Hugh, I couldn't resist. But you know, you've taken us through chemistry and physics and biology and a history of science so elegantly, and led on to some really exciting work I think that you're doing in the lab. And who'd have thought it that a bit of red light could actually have such profound biological effects on the eye? So absolutely fascinating, and I learned a lot as well from your talk there. You know, a reminder of some things, but also some really interesting new things as well. So we've got some questions rolling in, Bob, and I hope you've got a few more minutes that you can take some of those for us. So the first was you've sort of addressed already. So Jan had asked, can this treatment help with aging eye diseases such as macular degeneration? You've told us a little bit about, you know, macular degeneration, but where do you see this fitting in, I guess, with other treatments that are available for macular degeneration? And are you thinking of wet or dry or both?
Well, one of the beauties of it is its relative safety. So we feel it can come to clinical trials quite quickly. The advantage of the system we have, that we have the laser delivered system, which has I guess advantage and disadvantage. The massive advantage is that we can deliver an exact dose, if you like, and a radiance to the exact position on the retina, because the ophthalmologist is focusing the laser light on the retina and delivering the exact amount, as opposed to an LED therapy where the patient is in control of the light. So in terms of the AMD, though, we actually have a clinical trial planned, ethics approval almost through, I understand, to trial PBM in wet AMD, actually. So what we'll be assessing is its use as adjunct to anti-VEGFs, with the aim of seeing if it can, what's routinely done these days in Australia and around the world mostly, is what's called a treat and extend program, where patients receiving injections will receive injections and we will attempt to stretch out the interval between injections as much as possible. Some patients reaching 12 weeks, which tends to be the maximum, others not. What we would like to do is see if the laser can increase the proportion of patients that stretch out to 12 weeks. So that will happen this year.
Excellent. So another question from Francesca. Do you think low light therapy can boost healthy cells to help them work better? Or is this something which has an effect mainly on sick cells, I guess is the implication?
Well, that is a really interesting question. Could this improve contrast sensitivity in fighter pilots? I don't know. Maybe in totally healthy young retinas that are completely well oxygenated already, it might be a stretch to say that it could squeeze out more vision, because I imagine that healthy young retinas that are already receiving plenty of nutrients are already maxing out their vision. But a healthy retina that is in suboptimal conditions, maybe like a fighter pilot or pilots or some other conditions, maybe it could.
I'd love to know what effect it would have on contrast sensitivity in glaucoma, Bob. That's something you've shown you can influence metabolically through modulating glucose as well, and it'd be fantastic to look at that in more detail as well. So another question from Jackie. So do you think the light therapy will be a treat and extend type protocol like anti-VEGF treatment?
Well, the disadvantage of the system we have is that it needs a highly experienced user, an operator, to use the laser on someone's retina. So, and if it was required very frequently, it just wouldn't be practical, I don't think. So, what in the series of experiments we're running this year is to try and determine what is the actual frequency of delivery that's required to produce an effect. So, I don't know at this point.
Yeah. And another question from Jackie, is the vision improvement sustained after cessation of the treatment, or in retinitis pigmentosa, do you need to continue that weekly therapy to maintain the benefit?
Yeah, that's a great question as well. We found over time, as 6 months went by, basically the effect wore off. It diminished over the ensuing months. That's right. So you would, at some point, you would need more, but exactly how much, we don't know.
Really interesting question from Peter van Weingarten. So congratulations, Bob. I'm fascinated about what the evolutionary basis for photobiomodulation might be. Do you think it may be a remnant of the photosynthetic capacity of ancient bacteria that become the mitochondria of eukaryotic cells?
Well, yeah, it is interesting to kind of try and speculate on that. I don't quite see it that way, but it certainly makes sense that living, life on Earth, evolving under the sun, is going to react to light and electromagnetic radiation in various forms. So I think the 670 nanometer effect just happens to be the wavelength that excites the copper atom. It activates the copper atom at maximal activity. So I think that's just that would happen. I think no matter what planet you'd evolved on, probably, if provided you still had copper atoms in your cells. But the idea that it might have something to do with evolution is interesting. But I don't think so in this case.
Bob, a question from Cheryl. Can light therapy be done oneself? And I guess my follow-up question is, how does the intensity of light that you're using relate, compare to staring at a bike light or something, and is it intensity or duration or a combination of the two, or total energy that seems to be important? It's not
particularly intense, so it's very comfortable for the patient. So that it's delivered for 90 seconds actually. The patient, so with a lens on the eye, the way we delivered it at the slit lamp, with the patient focusing on the light. It's a 4 millimeter spot size, really large spot size, focused right on the fovea. Now people can deliver it themselves, but not with the system we have. Other researchers have developed light-emitting diode type systems like the eye patch that I mentioned. That was what the researchers from the US used. They used a system which had some LED lights on an eye patch, and the patients used it themselves at home.
Great. And another question of Jackie. Jackie's on fire this evening. So in the clinical trials, how do you blind the control group? And I think she means how do you mask the control group because obviously if the light is visible, they're going to know which group they're in. And does that affect their performance?
Yeah, that's a good question too. Yeah, hopefully we're not blinding the control group, but we're masking the control group. It's possible to set up a reasonable sham of this, provided the patients haven't definitely had genuine treatment beforehand. You can set up the slit lamp and a sham light that provides a reasonable type of sham.
Yeah,
we think. Yeah.
Interesting. And I guess a sort of related question from Shannon: is it possible to get a healthy dose of red light via sunlight on a daily basis?
Well, yes. There's the idea is that red light is good, sort of thing. It wouldn't be the dose that you receive just from walking around on a sunny day probably would be way less than required to actually have any sort of effect that we're talking about here.
Great, and I'm just seeing if there are any final questions. We're up to our hour of time now, so to be respectful of everybody's time, Bob, I'd just like to thank you for a really inspiring and thought-provoking lecture. I hesitate to say enlightening, but it's been a really eye-opening experience for us to see the effect of something that we're all familiar with, light, having such a profound effect potentially on the retina. So I'd like to invite everyone to join us in a virtual round of applause for Bob. So congratulations, Bob. Spectacular lecture. And thank you everyone for joining us tonight and for supporting CIRA and our research. If you have any further information that you would like to obtain about CIRA and the work we do, please visit cira.org.au. Bob, we have a gift for you. Believe it or not, it is stuck in Queensland, and that is not the excuse of the day. That is actually true. And so that will be finding its way to you when the logistic barriers and the awful situation in Queensland gets a bit better, and some of the goods are able to move again. So we hope to get a photograph of you with that when it arrives, and we will distribute that on our channels. To thank you once again for your lecture.
Thanks so much.
Thank you. We look forward to seeing many of our supporters and everyone in person through the year. And so, Bob, thanks again, and everyone enjoy the rest of your evening. Thanks for joining us, and bye for now.